Hello and welcome everyone to WebHub's Biotech Odyssey channel. And today in this video, we are going to discuss on the topic genetically modified organisms. So, let us begin. So, in this topic, we will be discuss the introduction and the techniques to develop the genetically modified organisms, the types of it, applications and example, and at last the overall conclusion. Right. So, what are genetically modified organisms, or what are transgenic animals? Right. So these are those organisms in which their DNA has been manipulated or altered through the genetic engineering techniques like recombinant DNA technology, CRISPR-Cas9, right, to introduce a new DNA or a new gene into their genome and obtaining the specific or the desired traits, right, from that organisms. Right. So the gene introduced will be called here as a transgene, and the organism formed through that transgene will be called as a transgenic animal. Right. So these are the genetically modified organisms. So moving on to the next part is your history and the timeline. So the first genetically modified plant is created in the year 1983, which was Nicotiana tabacum, and the first genetically modified animal created is a mouse, which has been created in the year 1974. So here, if we look at the brief history of the GMOs from starting from 1992 to 2021, some genetically modified organism has been created. Like here we can see the genetically modified corn, cotton, right, in there 1996, and the Arctic apples has been created with the technique called RNA interference in the year 2015, right. So this is the history of the genetic modified organisms, and moving on to the techniques to develop these genetically modified organisms. So the first technique is your recombinant DNA technology. So the recombinant DNA technology, we are going to combine the two different DNAs from the different species and create a new hybrid DNA or a new set of genes, right. So the process starts like firstly you have to identify your gene of interest. Right, you will identify your gene of interest and then you will isolate it by using the restriction endonuclease enzyme. This enzyme will cleave and isolate the gene of interest, and then after this, you have to transfer this gene into the host cell or the organism. Then you will need a vector to transfer it. So in that case, you will need a vector that will be a plasmid, and then you will insert the gene of interest into that plasmid, and then you will insert the plasmid containing the target gene of interest into the host cell or the target organism, right. So that's how the recombinant DNA technology works in the development of genetically modified organism. Now discuss on the second technique, that is CRISPR-Cas9. CRISPR means clustered regularly interspaced short palindromic repeats, which is a specific DNA sequences, right? And the Cas9 is your endonuclease enzyme, which will cleave the DNA at a specific position in the genome, right. So let me tell you that this technique is highly precise and accurate as compared to the RDT technique and upcoming other techniques, right. It is very precise and accurate, right. So as it prevents the off targeting, right. So the process starts like we will design a guide RNA, right, where that has a complementary sequence with that of a specific DNA sequences, right. And it will you can say the match with the specific DNA sequences, and then after it will guide the Cas9 enzyme at the specific location, and the Cas9 which is the endonuclease enzyme which will cleave the DNA at a specific position in the genome, right. And then after this, either the gene can be you can remove the gene, and in place of that you can insert your target gene, or you can simply also remove the gene, right? And then after this, the through homologous recombination, the DNA will be get repaired and the gene will be inserted, right? And the next uh, technique is your micro injection. So in micro injection, we will take a female egg cell and then we will fertilize it, right? After the fertilization, the zygote will be formed, or you can say we will take the fertilized egg and we will hold through it the holding pipette in the second step, and with the help of the micro injection containing the target DNA, right? And we will insert our target DNA into the fertilized egg, into the directly into the nucleus, right? And then after this, the fertilized egg, the zygote formed, will be inserted into the uh, reproductive tract or the uterus of the female animal. And then after this, uh, zygote or the transgenic zygote will be developed into a complete offspring, and the offspring or the organism formed will be a transgenic organism, right? Here in this case we have taken the mouse. We can also take other animals like sheep uh, and etc. So the other technique is agrobacterium mediated transformation, in which we will isolate the TI plasmid from the bacteria called Agrobacterium tumefaciens, and then with the help of restriction endonuclease we will cut the plasmid, right? And we will insert the foreign DNA or the target gene of interest into that plasmid, right, which will be acting as a vector, and it will form a recombinant plasmid as you can see over here, right? And then this recombinant TI plasmid, right, is then uh, reinserted into the bacteria, right? And then in the fourth step, you can see this plasmid containing the target gene of interest will be inserted into the plant cell, right? Plant cell chromosome. And then after this, these plant cells are cultured and it will develop into a transgenic plant that will show the expression of that target gene that we have inserted, right? So this is the techniques to develop the genetically modified organisms or the transgenic organisms. Now the next one is your types of genetically modified organisms. So the first type is your transgenic organism. These organism that contains the gene from the different species, right? And the example is Bt corn, that includes the gene from the bacteria which is called as Bacillus thuringiensis, that produce the toxin uh, that produces a protein that is toxic to the insect, that it gets activated in the alkaline pH in the gut of the insects, and it creates the pores which kills the insect, right? And the second one is your cisgenic organism. Cisgenic organism that contains the gene which has been transferred between the same species. The gene which has been transferred between the same species or approximately same species of the organisms. Right. The example is like apples engineered to resist diseases by introducing the genes from the other apple varieties. Right. The third one is your subgenic organism. These are organism in which their genes or their genes in their own state has been or you can say the existing gene has been altered and modified, right, without inserting any foreign DNA or transgene. You have to remember that in this case we are not going to insert any foreign DNA, right? The existing gene will be modified, right? So the example is like crops modified for drought tolerance by altering their existing genetic makeup. And the fourth one is your RNA interference. This technique is used in which for the silencing of the gene expression, or you want to silence a specific gene that encodes a harmful protein, or that you want to eliminate it, right? So RNA I use for the gene silencing process and to eliminate the expression for the certain genes. Like example, Arctic apples that don't brown due to silencing of polyphenol oxidase enzyme, right? So basically, the gene which codes for this polyphenol oxidase will be get silenced. Its expression gets silenced, and due to this, gene will not be able to code this enzyme, and the apples will not get brown, right? And the next one is your marker assisted selection. The organisms in which we are selecting the desired traits or the genes by using the biological markers or the DNA markers, right? For example, like crop bred for disease resistance are identified by the DNA marker selection. So, such type of is a marker assisted selection, right? Moving on to the applications and the examples. So, the first applications uh, in agriculture, you can see the in agriculture we have pest resistant, drought and salinity tolerance, and nutritional enhancement. Like in pest resistant, you are modifying the genome or the DNA of the plants that they get genetically resistant from the insect that destroys the crops, right? And you are making them unable to survive in the harsh environment, in the harsh conditions, right? Like in the absence of water or in extreme salt conditions, right? So, and the third one, nutrition enhancement, that you are increasing or you are adding some extra uh, nutritions to the plants by modifying their DNA and introducing a new gene to increase their nutritional value, right? That will benefit for the human welfare. And the second one is your medicine, like how so the first is vaccine development, right? You are going to create the vaccine, and the second is gene therapy. The third one is your pharmaceutical production. Means the you are creating such suppose example that you have created a genetically modified cow that produces the milk that contains a specific protein. Let's suppose human lactoferrin protein in its milk that boost the immune system, right? So that protein will be harvested from their milk and used for the development of the drugs that will enhance or that will boost or develop the immune system, right? So in the environmental applications, so what are the environmental applications? Like bioremediation, phytoremediation. In bioremediation, uh, we are genetically engineered the microbes to and plants as well. Phytoremediation, in plants as well, we are modifying the genes of the microbes and plants both, so to increase the efficiency of the biodegradation and to increase the capacity of their degradation, right? To eliminate the contaminants from the environment and to reduce the environmental pollution. So that's how we are going to implement the genetically modified organisms in agriculture, medicine, and environment. So next, the examples of the genetically modified examples. So the first one is your plants. In plants we have bt cotton and the golden rice, and in the animals we have genetically modified salmon and the cattle with enhanced milk production. And third one is your microbes that produce the insulin and biofuel producing microbes, right? So let me show you some the images of the genetically modified or genetically engineered organisms. So the first one is your genetically modified salmon. As you can see, the first fish, the upper fish, which is larger in size, this is a genetically modified salmon. As you know that normal salmon takes time to grow, right? So genetically modified salmon will grow larger in size and in a less time period, right, as compared to a normal one, right? And the next one is your monkey pig chimera. The second image you are seeing is a pig, right? It's a monkey pig chimera. And the third one is a vacanty mouse. You can see in the below that this mouse is containing a human ear on its back, right? Which is also an example of the genetically modified organism. So these are the genetically modified organism that has been created. Now, the conclusion that overall discussion about the genetically modified organism. These organism provides and offers us the advantage and benefits in to human welfare in agriculture, medicine, and environmental protection as well. Like in agriculture, as I told you earlier, that the plants, transgenic plants, which have the characteristics of pest resistant, that helps the plant to prevent them from the insect to destroy the plant, right? And they're containing the increased nutritional value. And in medicine, we are creating genetically modified cow. And now let's take another example in medicine, like genetically modified goats that contains the antithrombin protein in their milk, which can be harvested from their milk to treat the blood clotting disorders in the humans, right? And similarly, spider silk producing goats that produce the spider silk protein in their milk, which can be harvested from their milk. So we can create a durable medical searchers and textiles, right? Now, in environmental protection, like earlier, I have told that in bioremediation and phytoremediation, we are increasing the efficiency of the microbes and the plants to increase the biodegradation process and the capacity of the degradation of the contaminants, right? Eliminating the contaminants from the environment. And through genetic modifications, we are creating these uh, mm, genetically modified organisms. And also, they are giving one most benefit, that we are reduced dependence on chemical pesticides and herbicides. Like when you spread a chemical pesticide on a plant, it will also affect the crop, and the humans, the consumers which are consuming it, will also be get affected, right? So that's how GMOs are also reducing the need for the chemical pesticide, right? And also in animals, they are reducing the need for the antibiotics as well, right? So this is the complete information about the genetically modified organisms. So if you like the video, please subscribe, like, and share it. And if you like the concept, and please comment on it. And so this is all for today, guys. And thank you so much for watching the video.